Yeah.
Okay, so we're live. Hello, welcome everybody, and especially to my three very good friends who I really appreciate. And we're going to talk a little bit about trusting experts. What is an expert, and particularly in regards to COVID. So I'm going to allow everyone to introduce themselves: Sarah, Clive, and Annemarie, and explain why they're here. My name's Steve. And I'm going to talk a bit about trust and the Global Trust Project as well. So maybe, Annemarie, we start with you. Maybe introduce yourself to us and what you do, or what experience it is that you're bringing to the table.
Yeah, so I'm Annemarie. I've been a physical therapist for about 16 years. And then I've been practicing as a haptotherapist for the past 9 years, which is a little bit in between physical therapy and psychotherapy. And at the moment, I'm finishing my master degree in evidence-based practice in healthcare. I hope to finish it within 2 or 3 weeks now, and that would make me a clinical epidemiologist. And basically, I've shifted a lot in perspectives from like a normal, regular medical scientific view to a more complementary, holistic view, and back to science again. So I think I can relate a lot with the questions that people have.
Great, thank you. And for those that don't know, what is an epidemiologist?
Yeah, that's so funny because normally when I would say I'm studying epidemiology, people would be like, what? And now since COVID, this profession is kind of playing out publicly so people have a better understanding of it. And basically an epidemiologist studies disease and the spread of disease, and you use scientific methods to do that, like methodology for understanding the world, like asking questions about the how and the why and who. And a clinical epidemiologist is basically more, we use the same techniques, but we look more at the patient care perspective. So we study diagnosis, prognosis, intervention, and how you kind of apply the evidence to the individual patient.
Great, thank you very much. And Clive.
Good afternoon, Steve. So I did a medical degree many moons ago now, and during my medical training, I also did a degree in parasitology, and infectious diseases. I worked as a hospital doctor in the UK and in Australia for 6 years, and then I became a consultant in the pharmaceutical industry, and I've been advising pharma companies for the last 12 years or so about research and development of drugs and marketing and post-marketing surveillance.
Great, thank you very much, Clive. So Dr. Clive, right?
Yeah, that's right.
And a good friend, and somebody who, again, I can really say has the heart of people in mind. You know, really.
So, and I guess just to add, so I've also done a training in holistic counselling. So similar to Annemarie, it's always been quite interesting to me, the bridging of kind of the Western paradigm and Eastern paradigm together, and finding complementary therapies that work well together.
Thanks, Clive. And Sarah.
So I am not quite from the same background as the other two. I'm more into the lab science point of view. So for about 10 or 11 years, I've been a molecular biologist. I've got a master's degree in molecular biology and phylogeny. And then after that, I went into a more clinical setting from the perspective of infectious diseases. I worked as a lab scientist on a bunch of different diseases, and then I went on to do my PhD, which I'm currently busy with, and it's in infectious diseases in a hospital setting. In South Africa, so I've worked kind of in rural and also urban hospital, but government health hospitals, and we basically do clinical research into any infectious disease that involves mothers and children, mainly vaccine preventable diseases, but at the moment you know, a lot of things are on hold for COVID. So I'm not really researching COVID so much per se. I'm involved in some of the research, but I wouldn't say I'm an expert on COVID. But I am an expert in molecular testing and also the process of clinical trials, all of that kind of stuff.
Great, thank you very much. And so I'm, as I said, I'm the founder of the Global Trust Project, which is really working to bridge conversations with people, companies. I've got a technology background, so I'm here with 2 scientists and a doctor, and I've done all my training on the job, 25 years in the city in tech, similar to Clive and Annemarie. I've done different coaching qualifications and alternative qualifications. Like I'm a yoga instructor. I teach breath work, advanced breath work with people, and yeah, I've been spending my time in business and connecting people and ideas. So that's mainly what I do. So I'll talk briefly about trust as well, and really the conversation that I've been having, and now all 3 of us are in a chat group as well. The conversation has really been about why people don't trust experts. And I think something that's important to say is all 4 of us are not representing an organization. These are our own individual points of view. There's 3 highly experienced experts that are wanting to share their personal view around what they're seeing. And we can do this again, if this is of use to people, then we can cover particular questions that people can send us. Today we thought we'd have a discussion around trust and experts. Why, you know, Clive had a few things to say, Annemarie does, and obviously Sarah. And then Sarah would just cover testing particularly. So there'll be a presentation at the end with some slides where she'll just explain testing, the different types of testing, because that's one of the main things that comes up. And I think we should also just say it's a Saturday. It's Saturday, the 5th of December in 2020. So I know Clive will probably be watching the clock because he might get interrupted. We're just normal people, you know, that really want to see the best happen in the world. We all have our concerns and, you know, ideas, but ultimately science is what's important. So
I think just another important disclaimer, like I don't regard myself as a COVID expert per se, but I work every day interpreting data and reading clinical trials and working with science and scientific method. So that's where my expertise lie.
Yeah. And I mean, if we just touch on that briefly, Clive, because, you know, people do research, you know, they listen to a few experts and people that may be experts, like you're highly qualified. You've got a couple of degrees. You're a medical doctor, practicing in hospitals in the UK. You're eminently qualified to do things that I wouldn't have the vaguest idea to understand. And I read a lot, and yet you wouldn't put yourself in the same frame as Sarah, and yet you're very qualified to be able to comment on that. So I think talking about that is very interesting between us, like when people do research but they've watched a few YouTube videos and read a few articles.
So I'm no longer working in hospital medicine at the moment, just to clarify.
Yes, yeah, and that's kind of what I'm saying, right? You're staying in your lane.
But you know, there's obviously a difference between you know, someone who works as a lab scientist and researcher, and you know, someone who is a clinical doctor, for example, and someone like me who works in pharma. I can actually hear my little son in the background starting to wake up.
Okay.
My wife always warns me about my booming voice.
Fair enough. So Annemarie, maybe you can say a little bit about what, you know, there was some framing that we discussed before the call.
Yeah, and I just want to agree also with what Clive said. I have the same. I'm not a COVID expert, but have some relevant knowledge of, you know, related fields when it comes to evidence and research and searching for it, appraising it, applying it, which is still very valuable when we're having a general discussion about expertise and not necessarily zoomed in on COVID. And as a framework, I think, you founded the Global Trust Project. So you're obviously invested in trust and bringing more trust in the world for it to kind of function better for us humans. And I think we all signal in a different way that there is a bit of a lack of trust in expertise and information, in science, and that it kind of leads us into things that we don't want, like, for instance, undermining public health measures or just basic unnecessary suffering. Part of it, I think, is a lack of information, or like a misunderstanding of information, and maybe just some people also have a higher propensity to kind of mistrust. So this is why we bring these questions to the table, that we've seen this come up on our timeline and have had discussions with friends who have these questions. And then things become more specific around COVID, of course, because that's right now the situation we are all in our different regions in the world. So I think that's what we're trying to do today.
Yeah, great. So should we kick it over to Sarah, maybe to respond to a little bit about what you're seeing, Sarah, and we can jump straight into the testing conversation. I know Clive had also some framing aspects as well, but Sarah, maybe give your comments. What are you seeing?
So I think for a couple of years, I've been very concerned about the level of mistrust, not only in government and science and pharma companies, but just the motives behind a lot of things. And it's kind of been playing out, mainly in mothers of young children when vaccines come up. But now with COVID, what we've seen is a complete amplification of all this. And like you said, some people will say, they're an expert in something, but they'll be very vague and say, I'm a doctor, or I am a scientist. But there's a lot of specific areas of science, and staying in your lane means I'm not an expert if somebody asks me about astrophysics, if somebody asks me about my medical opinion on something, I can't give that. I'm not a doctor. I'm not trained to do that. And I think what Annemarie was also saying earlier is that, there's got to be the hierarchy of evidence that we've got to look at. And at the end of the day, we're a couple of people here who are all experts in different things, but expert opinion is the lowest form of evidence. So if anything I'm saying today, or Clive or Annemarie or Steve is saying, goes against the consensus of science or the consensus of medicine, you should be interrogating the higher forms of evidence, like systematic reviews, the published literature, and not taking expert opinion as the end form of evidence. So I think from my point of view, I've just been trying to really explain to people the meaning behind the science, so that they can kind of look at the evidence that's there and actually understand it.
Annemarie, any comments on that?
Yeah, I really agree. Like, from a perspective of, there's science, but of course some science is better than other science. And I think that's a discernment that I couldn't make before I was actually trained to look at it in that way. I just thought, someone's an expert, so they have something to say. They know more than me, so it must be right. And the interesting thing in the scientific process is that you're always looking at what you're missing. So if you have one expert opinion of someone who is saying one thing, you kind of have to systematically look at, okay, but what is other information that we need that kind of puts this into perspective? And I think that's a thing that's come up a lot in social media and in videos, that someone presents their expert opinion and their evidence, and people actually have no idea if any other experts have very contradictory views, or if this expert is saying something that is against the consensus, or if maybe the things that they've been saying, that there's evidence against it. And I think that's really one of the things that you're trying to do when you look at something in a scientific way, is you're not looking to confirm your question or to just trust someone saying something, but you have to kind of dig deeper, and there's a way for doing that. So that's definitely, yeah, and like the very aggregated forms of evidence where just all kinds of studies are being combined into one conclusion, like the systematic reviews, what Sarah said, like they're on top of the evidence hierarchy, like the evidence pyramid. Ideally, you would have those, but they're not always there. Yeah.
And I think what's interesting for me is that the average person doesn't know how to do that. And, you know, even me, I talk to the three of you quite regularly. I field a lot of questions from people about COVID. And then I come and ask you, Sarah, I don't know how she's so patient with me. I'm always asking her. Clive and I trade messages, Annemarie and I have, and lots of my other friends who, you know, are experts in their particular area. And even then, it's about, how do you trust, which experts do you trust, and how do you come up with the body of evidence? And there's a great article that was released in Forbes about before you question an expert, things to do, and what doing your own research is compared to actual research. So Clive, you were coming off mute. I think you had something to add.
No, I did. It was just about you know, the experience I've been having of late with friends and some of the conversations that we've been having. You know, people get their information in so many different ways now, and the majority of sources tend to be, you know, Googling something and reading a very long and detailed article, which usually is like a second-hand report, sometimes of clinical data, sometimes just opinion. And you know, the people. So what Annemarie was referring to was, you know, the being able to critically appraise the source material of a trial, of a study. And I think, you know, in order to have the skill to do that, you need training, and it's practice. You just have to practice looking at clinical data and understanding the limitations of it. And just because something's written in a trial, you know, doesn't necessarily mean it was correct. You need to understand the ins and outs of the trial. So, you know, I've certainly been finding that, and a good example is in the vaccine research that's just come out. You know, people are making all sorts of claims about the studies that have happened, and I've been asked, you know, but you know, what have the studies shown? And I think that's the key part of it, is that you know, you do need experts to be able to interpret data in the correct way, in this way. And then there's the question that you were alluding to, Steve, which is the Forbes article. But each expert could have a different way of interpreting data. So then you need to have your skeptical hat on to know which experts you want to listen to. What are the right questions for the experts?
But then there's this thing about general consensus. So even though the majority of experts might disagree, or, you know, some experts might disagree with something, there is a majority consensus about scientific principles. And I think that's really important. And again, Sarah, maybe you can touch on that when you go through your piece. I'm conscious about time and how we try and make this, you know, impactful for people. So I'm just very briefly going to touch on trust. And I'll just share some slides here, and then we'll kick it over to Sarah to share her slides. So I often give this presentation about the body of trust, and I talk about trust as being contextual. So trust really is something that is different for different people in different circumstances. And trust in business can be correlated to business metrics. I then go on and I ask people, what is trust? And it's very difficult for people to define what trust is without using the word trust. So again, I normally take about a half an hour to 45 minutes to give this presentation, which I'm not going to do. But the science of interpersonal trust says that trust is the willingness to accept vulnerability or risk based on the expectation of another person's behavior. And that's a really densely packed sentence with lots of juicy things in there around vulnerability, risk, and it's about benevolence. How do I feel about the British government towards me, the South African government towards me? How do I feel about scientists? Do I feel that they're being paid by a farmer and so I can't trust them? So, do they have integrity? Do they have the capability to do what they say they're going to do? And that's from the science of trust. And there's some real data, 50 years worth of metadata, and the literature, which is a word that, again, most people don't fully understand the depth of just the meaning of the word literature. But from the literature, trust comes down to three things: ability or capability. So do these three people here, these two scientists and this doctor, have the ability to interpret things in a way that is useful to me as an individual? Do they have integrity, or are they lying? And again, I can vouch for the three of them, but people have to get to know people, say, yeah, well, is there integrity? And is there benevolence? Do they have my best interests at heart? And that's really what trust is. And when we answer that, when we really come back to the root of trust, which is what the Trust Project is about, then we can start to change the world. And the answer is action based on trust. And we've lost Sarah. I know her internet is pretty poor over there in South Africa. So I'll stop the presentation, and yeah, we may have to pause for a bit. And yeah, let me just press pause and see. So Sarah's in South Africa. She's got a storm. We were just having a chat as we pressed pause. She's back, and Clive was just saying, his son's woken up, and then we're having this relaxed conversation, and we were talking about how experts sometimes aren't relatable. So Clive, yeah, just say what you were saying, Clive. I think that's perfect.
Yeah, it's just that. It's using language that isn't accessible to people. I guess it's interesting about the underlying intentions of people and trying to gauge that when you listen to them. Our intention here is to build trust, and is to help people to be able to discern the fact from fiction in a way.
There you go. And it's personal, right? I mean, all of us have got our own personal lives going on. I mean, Sarah just mentioned before about a personal situation, relating to COVID, right? Clive, you were just saying your son's woken up, your wife is over there, you've just had COVID, right? Again, I don't want to give away anyone's personal sort of information, but
I think it's true. All these things come with a load. They're not just it's not just a mental process. It's always, there's a lot of emotion that comes up with these, especially with COVID and how much it's affected people's lives. And I think it's kind of having discussions where the head is engaged and also the heart is engaged, I think, and perhaps that's sometimes what's missing in these conversations.
Yeah.
Steve, if I could just say, because you alluded to my personal situation, and I really don't mind talking about it, but like you're saying, Clive, with actually realizing that there's real people behind things. And I know for myself, this week, I've really realized the difference between seeing on TV that someone's got COVID and the family's not allowed to go in and say goodbye to them or speak to them, and they're kind of alone in a hospital room, and you don't really prepare yourself for how hard that is until you're in that situation. And I'm in that situation in the moment. My grand's in hospital. She caught COVID when she was already sick, and no one can go and comfort her. No one can go and tell her, even though she's so scared at the moment, that, we're there for her. We're all rooting for her. And it's affecting my family more than I ever thought that it would. My mom's usually a really strong person, and she's struggling with not being able to actually go and sit with her mom and help her through this. And that's what everyone's going through. You know, we're all going through this thing, and everyone's got a personal story. I've got friends who, you know, use their brain for their work, and they've got, you know, the whole long COVID where they're not able to think. So
yeah, it's tough. And Annemarie, you were saying before as well, just some of the things that you're seeing in terms of trust and how people feel. I mean, it's a lot about feeling, you were saying beforehand.
Yeah, and I think, like, because everyone has this personal story and their personal context that they're finding themselves in, you know, in your situation, Sarah, wouldn't you just wish that COVID didn't exist and that the measures weren't there so that your mom could be with her mom and help her? But you understand, you know, more of the reasoning behind it, so you can kind of park that what you would want to do short term, and kind of understand why this is the case. And I think that's, you know, just having a broader perspective. But many people are in their own, looking from their own perspective, and think like, you know, lift the measures and my issue will be solved, in a way. So it is like so human and so understandable that we have these wishes and we have these needs and we have these longings, and also, you know, I think we do actually need to question things. That's a good thing that needs to happen. Like, these people should be there and they should be heard and have a voice. But yeah, I think that's really a thing. And like I said, I think some people are just have more of a default setting of mistrusting institutions, science, authority, experts, and in many cases that's also because they have bad experiences with it, or they've not been seen in interactions with medical professionals, or haven't felt that they were rightly understood, or have felt like there was a protocol placed on top of them instead of their personal values having been respected in that. And I think that that's also contributing to this in a way. Yeah.
I would really agree with that. Sorry, Steve. And the other thing I was just thinking about is, and this is a journey that a personal journey that I've been on about, you know, at the beginning of the pandemic, when it first started, I immersed myself in the data, you know, every day. And because, you know, but last year there was so much was happening in such a short period of time, and that there was so much we didn't understand and still don't actually. But you know, I was sort of reading voraciously and really trying to understand, to the point where, and like commenting a lot on social media and trying to draw out, you know, what I thought were the facts from fiction. And I got really like super saturated, and I couldn't actually do it for very long. It became quite overwhelming because of the depth and the level of uncertainty and the conflicting data that was coming out. So, you know, I found I work with data, and I work with clinical papers, and I was finding it completely overwhelming and you know, very difficult to stay on top of. So, you know, I think I really understand it as a coping strategy to want to know, to not want to sit in that uncertainty. And I think one of the consequences of wanting to know and not sitting in that uncertainty is maybe being drawn down these kind of, I guess they're like rabbit holes, I suppose. They don't really lead anywhere. But, you know, this propensity to be drawn in, and then you can keep, and this is one of the problems with social media and with with Google is that, you know, confirmation bias is such a thing that you can keep looking things up and further reinforcing your beliefs about something, and until you then become very fixed in your belief that this particular, whatever, I don't want to use the word conspiracy, but just that you know, you're just constantly reinforcing what you already believe.
What you think you know. Yeah. It's such a valid point. And the other thing I think is, I'm just conscious, you know, we planned this, we wanted to share really crisp information, and you're kind of competing with, you know, very slick, very professional YouTube creations and people that have got an agenda, that are just, you know, pushing a particular thing, splicing it up, and not having honest conversations. And so again, you know, for me, it's about people taking the time, you know, to watch an hour recording where they get to know us and really understand, you know, who is really saying what they're saying. You know, you as two scientists and a doctor, and you're not speaking for your organizations, but within your professional ability and capacity to say, this is what we're seeing out here. And I think that's really important. So, you know, with that, maybe Sarah, we can turn it over to you and your cat. And let's just talk about testing. So, you know, testing is a big thing. You know, so you've got some slides which you're going to go through. So maybe before I put the slides up, and then and you talk us through them, maybe tell us what it is that you're going to talk to us about, and why. Why is this important?
So I think, Steve, a lot of the voice notes that I've been getting from people and videos that people have been sending me about asking my opinion have been about this whole false positive paradigm in the testing of samples. How do we determine what's positive for COVID? And I think that's kind of what I was hoping to address with this PowerPoint presentation. And a couple of people also say things like the test for COVID detects all sorts of other respiratory viruses, and that's not quite the case. And I know some people who are very good scientists who have been talking about lateral flow antigen testing and how they've got a much bigger advantage over PCR testing, have been saying PCR tests have a lot of false positives. And people are taking that to mean that it's detecting the virus when it's not there. And that's not quite what these scientists are meaning. And so different tests, I guess, have different applications, sorry. And, you know, a lateral flow antigen test is very useful for telling you if you're infectious now. And I'm kind of going to go through a bit and explain why it has that advantage over PCR and what the advantages of PCR are over a lateral flow test. So yeah, if you're ready to start, we can go through it.
I can start it up. So there you go. So what are false positives in PCR testing for COVID? And does the test detect other viruses, including flu or other coronaviruses, right? So you just tell me when you want me to slide across.
Okay, so while I'm talking about PCR, obviously, you have to know some of the concepts that I'm talking about. So just to make sure everyone's on the same page, I just wanted to show you this. This is a simplified annotation of the SARS-CoV-2 virus that causes COVID, and the triangles are the spikes that they refer to that have a receptor binding domain. This is complementary to a site on our own cells, and this is basically what the virus uses to attach to our cells and enter our cells. And this is also where antibodies would attach and then block this binding. So they prevent the virus from being able to infect our cells. So in yellow, I'm showing that the virus has RNA, which is held within a nucleocapsid. The RNA is basically instructions to code for all the viral proteins, like the spike, for example. So when it's in our cells, the virus's RNA instructs our cells to make those proteins and construct more virus. So it's these instructions that we're actually targeting with PCR, the RNA. When you have an antigen test, you're targeting the actual proteins of the virus, so those triangles and not the yellow. And so when you're looking for a serological test, you're looking at the antibodies which bind to the virus. So this is how all these different tests work. So if we can go to the next slide.
And I mean, Clive or Annemarie, do you want to comment at all, or should we just run through the slides? You know, if you guys want to jump in, then and feel free.
Okay.
Okay.
Yeah, so when we do a PCR, we're looking for that RNA. So we take a sample from a place in the body that we expect the virus to be, for example, the nose and throat, and then we perform an RNA extraction, which takes out just the RNA and gets rid of all the proteins and other things. And then because polymerase, which is the enzyme that they use in PCR to copy the DNA, is looking for DNA, we basically use a reverse transcription to make cDNA from RNA. And then you do the PCR. So that's what this slide is showing here. The PCR is basically a set of enzymes that make multiple copies of a target piece of DNA, just like a photocopier, but using temperature cycles, and with each cycle, exponential copies of that target region are made. So that's what I'm showing in blue. So the purple is kind of the whole RNA sequence or the whole DNA sequence. And with PCR, you're just making a copy of a target region. Now, how PCR works, especially real-time PCR that most people are using at the moment, is you have a forward and reverse primer, which are shown in blue. And those attach to the target region that you're trying to copy. And they'll only attach if that target region is a match. And then you have a probe, which is even more specific, and that also has to match. So it's got a fluorophore and a quencher, and when they're in close proximity to each other, there's no fluorescence. But when you separate the two from each other, then you get fluorescence. And so as the PCR proceeds, basically the primers attach, the probe attaches, and then you get the polymerase copying the DNA. That chews up the probe, which then separates the fluorophore from the quencher, and you get fluorescence. And the more copies there are of the target region, the more fluorescence you get. And so you can actually measure this.
So,
Sorry,
I mean,
skipped the next slide. I forgot to say next slide.
Oh, right. Yeah, okay.
You lost already, Steve.
Yeah, right. This is it, exactly, right? So, you know, for Clive and Annemarie, you know, for the, like, it gets so complicated so quick, you know, we probably need some, like, slide guru to help. But what did we just...
Can I say something about that?
Yeah.
Because I was like exactly thinking that, like listening to Sarah and thinking like, ooh, you know, I'm quite smart in these things and I can follow, but it is already kind of hard to keep your attention with it. So imagine if you're just, you know, a random person and you have to understand it. And I think this is really interesting. Like, how do we translate science to people so that they understand without losing really important information? Because simplification is also something you don't always want. So I was listening to it and kind of parallel processing. I think I can sort of summarize it in normal language. And I'll have a go with it.
So this slide is actually more helpful for what I was talking about, because I was actually referring to this slide. I was trying to look at the PowerPoint on my own screen.
Even then, Sarah.
Yeah. But I think if you would want to translate it, it's like if you take a swab and there are cells in it, they contain the RNA, but that's like kind of a building like a construction template for new viruses, right?
Yes.
So that's like the instruction of how to make more virus. And you kind of negative copy that. Like you can make from a photograph, you can make like a negative. And then if you have a swab and you do the PCR test, that negative that we've copied kind of turns up fluorescent. And then you can see that the actual building blocks for the virus were in your swab. Is that it?
Yeah, sort of. So yeah, this slide here, right? The purple is your RNA, the blue is your target region. You get your two primers that attach. So you're only copying a small portion of it, a fragment of the instructions, only for the specific region. the RNA contains the instructions for the whole virus. We're just interested in the nuclear capsid or just the spike or just somewhere that's unique to this virus.
Yeah, so we have page 15 from our IKEA building kit. And then we know exactly which, closet we're putting together because this is unique to that closet we're building.
Exactly.
Yeah, great. Okay.
And from a, I guess from a clinical application standpoint, the thing that makes, well, my understanding of PCR and the thing that makes it so useful is that you can detect tiny amounts of viral genetic material. And by using the PCR test, you even if you've got tiny amounts present in the body, you use this process to amplify tiny amounts into long enough strands that you can then detect, right? Is that right, Sarah?
Not long enough, lots of copies of the same thing. Yeah.
So you can then analyze them and see if it's present.
Exactly.
And fluorescing. So fluorescing means what? Because people are going to get stuck on fluorescing. I mean, I got stuck on just about every word you said. Yeah, no, and you've explained it to me before. Like the one voice note I've starred and forwarded to people only because it's like, guys, you understand how complex it is, what everyone's working on. And I think that's what's so nice. There's 3 experts and one expert is explained to 2 other really serious experts. And they're like, so pretty much I get what you're saying. And, so yeah, what's fluorescing?
Fluorescing is like, I suppose, releasing light energy.
And you can
actually
like a petri dish. So you're able to see it. It's fluorescing.
Well, you can't see it, but the machine can see it. It's a lot tinier than anything you can see with your naked eye.
Right.
You can sometimes see it under a microscope, but yeah.
You do this. I mean, one of the voice notes you sent to me is, this is really easy. I'll just, guy had a question and you're like, I'll just send him the thing. He can look at the public data. But that's the point, right? This is all publicly available. It's not like secret squirrels, or hidden away. This is like anybody who takes the time to get 3 or 4 degrees like you have could go and do this, right?
But I think we're touching upon like a very key point that when you get to for looking at a subject like molecular biology, which Sarah is an expert in, the science of molecular biology is complicated. So you need experts to be able to explain that data to you in a way that you can understand. And this is where the problem keeps arising, because it's like, which experts do you choose? And and the technologies and the science that underlies some of these processes, some of them are complicated, when you drill down,
right? Like, who's paying for all these labs? Where's the money coming from? So just once you start, I mean, I always say this, people don't understand billions. They think they do because everyone talks about it in billions, but very few people actually really truly understand what a billion of anything is. A billion tests, a billion people, a billion viruses, anyway, big numbers. So Sarah.
I think questions that people could have right now is like, are you sure the mini part of RNA that you're copying is actually from COVID, or could it be of something else that looks like it? And if you make like a super amount of copies of something, could it pick it up while it's not actually really there?
So I think that's what we definitely go through towards the end. And I'm kind of trying to teach you a little bit of the basics here so that when we go through that, it makes sense.
Thanks.
So, so basically, at the end of the day, your question is exactly what we're trying to address here. Great. With specifics. So yeah, let's go back through this quickly. So basically, we're looking for only the RNA, the instructions. We're taking a very specific piece that we're interested in that we know is unique to the virus. And how do we know that? We'll go through that in a little while. And we're making multiple copies of that. And while it's making copies, it's kind of producing a bit of fluorescence that we can detect. Now, primers and probes only attach to a direct match. There can be one or two mismatches, but it has to be pretty much the exact thing it's looking for. Okay, Steve. So here in these cycles at the bottom that I'm showing is just how it's like taking something. I think you skipped, yeah. So it's basically taking something, making a bunch of copies of it, and as it makes copies of it, it's releasing light, and we can measure that light. And this is basically what a PCR graph will usually look like. As there's more copies of it, it releases more light. So, and that light, if it's produced sooner, if it detects the multiple copies early on in the cycles, then that's the first arrow that I show at cycle number 12. It'll release a lot of fluorescence early on. If there's less virus or less copies of what you're looking for, it releases it later on. So say at cycle 26, or if there's very few copies at cycle 38. So generally after cycle 40, we say it's negative. If it wasn't detected before 40 cycles, it's negative. Because it's exponential, there's obviously so few copies of it that we can't detect it. So yeah, if you could go to the next slide. So with that in mind, I've basically proposed a hypothetical situation here where we're looking at days since infection and viral copies. Now, viral copies, the more copies there are, the sooner it shows up on the graph, and the lower the CT number. Okay, so remember, the more virus, the earlier it comes up. So the less virus, it might not be detected. So the darker blue here kind of shows, I guess, the viral copies. And as those go up, the increasing likelihood that you're actually going to detect it. So
people might have a question. Well, something that's going through my head is, and maybe we don't know the answer to this, but it's how much exposure of virus would you need to get infected from somebody, for example? So because lots of people talk about viral load, and that's supposing someone who was infectious with COVID coughed on you, what level of virus are we talking about, just to give some sort of context to the we're talking about viral copies and amplifying that in the test, but what kind of numbers typically would we be talking about?
Oh, I don't know those numbers off the top of my head, Clive, but I think it's early on when I looked, it was something like 40 to 1,000 copies of the virus.
Right, okay.
But here, this is not really showing how much you're exposed to. This is showing someone's course of infection.
Right, okay.
So days since infection and what your viral load is inherently. So that's more what I'm trying to show here. So it's very hard to actually measure what you're talking about in terms of how many viral copies or viruses, individual viruses it takes to establish an infection. That's very difficult to kind of quantify.
The reason why I ask is just that certainly from some of the case reports I've read, the people that tended to get more severe infection tended to be those that were exposed to a higher viral load. So we know that, but it's I guess the question is more, what's the minimum viral load that you would need to have in your system before you could detect it with PCR?
Well, PCR is very sensitive. It can detect about, I suppose, if you bear in mind, when I'm talking about copies, I'm talking about what's in the swab, not necessarily what's in your body.
Right.
So in a swab, it can detect, 1,000 to 1,000,000 copies. It can detect that. Down to 100 copies, it can detect that. So down to 100 individual viruses, that's been taken by the swab, and that is in the vial that you've extracted. So yeah, it is very sensitive.
Yeah, it brings up lots of questions, but okay, so next slide?
No, wait.
Oh, okay.
1 second. Let me just get back to where we were. Yeah, okay, you can go to the next slide.
Okay.
So in terms of infectiousness, the WHO basically says you're most infectious 1 to 2 days before symptoms, and symptoms usually start on an average day 5. Okay, so that's that pink bar that I'm showing at the bottom there that says infectiousness. And you remain infectious probably for around 8 days after your symptoms start. So after that, you start to form antibodies, your immune cells start to kick in, and it starts to actually destroy the virus. And a lot of how it does this is by breaking it up. But you still have low copies of virus after this infectious time point. So after the pink bar starts to disappear, you can still detect the virus, even if it's just fragments of its nucleic acid, its RNA, its instructions. Even if it's not instructions for the whole virus, you can still detect it with PCR. So if you skip ahead to the next slide, Steve. So in green, I've put basically the possibility that you would detect it with PCR based on the CT value, that value where it starts to fluoresce enough that you can actually detect it. So even though there's only still remnants of the virus and you're not still infectious, as you can see, the green bar is a lot longer than the pink bar. So does that make sense?
Yeah, that makes sense, yeah.
So if you then skip ahead to the next slide, so that's where the false positives come in. In terms of actual infectiousness, you might no longer be infectious and you might not even be infected, or the virus may be dead, but it would still show up as positive on a PCR test. And this is what they call a false positive for infectiousness or being infected. So when I'm talking about a false positive, usually this is what we talk about in terms of PCR. Okay. So if we could skip to the next slide. So this is where the antigen test comes in. It's not as sensitive as PCR. It'll only pick it up when you've got a higher number of viral copies. So that's the yellow arrow that I'm showing. And the viral load has to be quite high for it to be detected on an antigen test, a lateral flow test. And so this is why it's a better test of infectiousness. Is that clear from the slide to you guys?
Yes.
So basically, that's where a PCR false positive comes in compared to a lateral flow test. So
just to explain what a lateral flow test compared to a PCR is.
Oh, yes, definitely. Thanks, Clive.
That's right.
Who's going to do that?
Well, Sarah, I was hoping.
I thought you were saying you were going to explain.
I mean, I can a bit, but I think you'd be able to explain it better.
Yeah, so basically a lateral flow test is essentially a piece of paper that's absorbative, and on the piece of paper, it's basically blotted with something that would react to one of the proteins that's in the virus and that's unique to the virus. And so when you put a swab or something onto the piece of paper and then you put a buffer on there, basically a line or two lines would show up, basically saying that there's been a very basic chemical reaction.
So Clive's showing that. Clive, you might have to say something how, depending on how it's recording.
So this is a lateral flow testing kit that I happen to have. They basically look like this. So it's
almost like you're a doctor, Clive.
It's all unbranded, but you can see here that you put a drop of blood in there, and then you put a couple of drops of buffer solution in here, and then depending on, so that it basically says C for control, and then underneath is the two antibodies that it's detecting. So it's detecting.
Yeah, so that's a serological antigen test.
Yeah.
So that's a completely, I mean, I'm showing the yellow arrow here. I'm talking about a lateral flow antigen test. So it's actually looking for the virus itself. The test that you're showing is basically looking for the antibodies, and those would show up basically beyond the region of this graph, because they kind of show up maybe a couple of days to a week or two after you're infected.
But they would both look similar to
exactly
to that plastic box with a place to drop your blood in and all the rest of it.
A blood or a nasal swab or whatever. Yeah.
Real life
Steve, do you mind if we paused for a second?
Yeah, let's do that. Recording. Okay. Yeah, so we're back. Sarah went to get some power. Clive's son banged his head, was yelling in the background, pushing around a cardboard box. Like real life was happening. And we were just again discussing how difficult it is to convey very complex problems simply. Right, Clive, you were just saying that.
Being distracted in the background. Yeah, I mean it's and I think it's in the language and I think in the words that are chosen, that if people don't understand the word, then it's then necessary to stop and explain what the word means, and it then takes a long time to explain.
That's why these simplifications happen, so. And then people get lost in the simplifications.
Yeah.
You need the words, which is why you need experts, because you studied all the words. But yeah. Anyway, Sarah,
it's great having you guys stop me and saying, "Hey, whoa, wait a second, that's not making sense."
Yeah.
Well, it makes sense, but it's just I think that it's like I said, it's the words that people won't be familiar with, that make it inaccessible.
Mm.
Yeah, we'll come back. We'll come back and do this in again, I think. But yeah, Sarah, why don't you carry on then in terms of what should I go to the next slide or?
I just want to quickly sum up what I was busy saying. So, you know, a PCR is making multiple copies. As you get more copies, there's more fluorescence. The infectiousness is well detected by a lateral flow antigen test, but beyond the point where you're infectious, it can still be detected by PCR, because PCR is very sensitive. It can still pick up very small quantities and also broken up pieces of virus. So I think, Steve, this is where I'd sent you a voice note once explaining it and saying, you know, if I come to your house and visit you and then I leave, and somebody comes into your house and says, well, was Sarah here? Picks up a strand of my hair, tests it for my DNA and says, well, she was here. You don't know, am I still here or was I there half an hour ago? So that's where PCR kind of, I guess that's where you get the false positives. But I was still there at the end of the day.
And that's the key. You're right. So people are misunderstanding what they mean by false positives, because they think they're saying false positive in terms of infectiousness. Is Sarah here right now versus was Sarah present at some point?
But it doesn't mean that, for example, from that hair that Annemarie came to visit you. No, it's specific to my hair.
Right.
And that's kind of what I wanted to take you guys through. How do we know that?
Okay, great. You can't visit me and we're in lockdown.
Yeah, well,
and you're in the wrong country.
Why would there be broken pieces of virus in the body?
So basically, as the body starts to recognize that there's proteins being made based on the instructions of some RNA that's not ours, the body basically signals those cells for destruction and starts to recognize those proteins and starts to actually attack the virus and break it up. So that's basically why there would be RNA kind of left over. And also, as you know, the immune system kicks in, your viral load decreases.
Right.
So,
yeah, so
do I understand it correctly that as the days since infection go up, the false positive rate goes up? Or is it like, because I know the sensitivity of a PCR test is around like 98%. Is that like, does it capture the whole time frame, or is it different on different times since infection?
Yeah, so that's
a technical question. I understand, but
5 days from infection is when you start to show symptoms. You start to show symptoms kind of when the load is going up and starting to have an effect on your body, and your body's starting to recognize it and respond. And then as your body responds, the viral load goes down again. until it eventually tapers off and you can't really detect the virus. I mean, some people have shown you can detect it at up to 21 days since the first day of infection. So, but that doesn't mean that you're infectious still.
I've heard even 3 months in videos from people who want to bash the PCR test.
Yes.
But so again, the lateral flow test also has a false positive rate, right? Like everything has a false positive rate, or not?
So the false positive rate with the antigen test is a little bit different. So where the antigen test is useful is it's more likely, because it's less sensitive, it doesn't pick up the virus for as long. It's more likely to tell you if you're infected today, but it won't tell you if you were infected a couple of days ago. And then they're not quite as specific as the PCR test. So sometimes they can pick up something else, but it's not a high amount. The problem with them more is that they're less sensitive. So if you've got low viral copies, so say, for example, before day 5, you may still be infected and infectious and they won't pick it up, but they're not going to pick it up at day 21 for sure. So where people say, you know, there's a fake epidemic, a testing epidemic, etc., of false positives, it's not that these cases don't represent people who have been infected. It's that perhaps they're not infectious right now.
Right.
So I hope that that's kind of clear.
I think that's, that is clear. And I think that's a really important point. And one of the things that's come up in discussions that I've had with some people is that, you know, clinically, you would never advise anyone on the basis of just a test. You know, in medicine, you would look at the patient, you look at their symptoms, you know, it depends. Pardon?
A case definition, essentially, plus the diagnostic results.
Yeah, so you have all your information in front of you with the PCR. Like, you never have a PCR on its own, and it's a corroborative piece of evidence.
Nice. Thanks.
But the complex thing about COVID-19 is that you can still transmit when you're infectious. So that's where the PCR tests have been kind of helpful. But they're not helpful in telling you if you're infectious.
Right.
So yeah, we ready to go to the next slide? Okay, so this is a whole lot of letters on a screen. I'm gonna try and explain it as best as I can. So this is the publicly available CDC assay set. So basically, assay set is the primers and the probes I was talking about earlier, basically the kits to copy the RNA that's publicly available on their website for SARS-CoV-2. So when I was preparing this, I went to their website, I copied this off. So there in blue are the primers. Those basically attach to the start of the region that you're looking for and the end of the region. The probe touches on a specific part in the middle. And what's in red is the sequence of the probe. So each letter is basically a base of RNA. RNA is made up of building blocks. So it's one of those little building blocks. And then basically what's in purple are the dyes and the fluorophores and the quenchers that I was talking about earlier. So let's skip past all that and let's just look at the probe. And you can do this for the primers and the probe, bearing in mind that they have to be a match in order for the reaction to proceed and for you to detect the fluorescence. Okay, so how do we know if these detect other pathogens?
I'm being annoying again and interrupting you again, and I'm really sorry because I keep interrupting your flow. But it's just because I know what Steve was saying, we were going to use this as like a kind of a practice recording that we can do, but something that just occurred to me. We could use some of it. Yeah, so we can use some of it. Yeah. But so what just popped into my head is that you have to aim at, you have to decide what level of scientific knowledge your listeners have. So if we're saying people, you know, have some science background, then these things make a lot more sense. And that's I think at the beginning, maybe we need to make some sort of decision about what level we're aiming it at, and take people up from, because maybe some people may not know what DNA is, because I've had that question about what is DNA, and that it might be that level that they don't have, that they're, and I think part of this discussion is about, and this has come up too, is about scientific literacy, and what does that actually mean. And a lot of the discussions I have with people are people who haven't studied science, and so if we're then teaching them about molecular biology, it's going to go over their head. So I think maybe there's a very quick way to take people from no knowledge to then get increasing complexity. I don't know, or maybe we have to state up front, this is for people who have a basic level of knowledge of DNA and of biology, and it was just something that popped into my head. So
yeah, I think you would have to assume that the people who kind of are looking up conspiracy theories, or looking up alternative theories, or questioning the science, are kind of people who are reading and trying to figure out what DNA is and what RNA is and what a PCR test is.
They do, yeah. but a lot of them don't have a scientific background. They're interested and they read, but I think this is where the difficulty sometimes arises, and that there's a kind of there's like an almost like, but I'm a smart person, and I'm smart in my own fields. Like, I don't need you to tell me, I can read it myself. But there's still a level of illiteracy, scientific illiteracy, I would say.
I think that's really important, because like even so we've been backwards and forwards. I've been fielding for 7 months different requests for information based on theories that are out there, and some of them are, again, like Clive, I don't want to use conspiracy theory as a word, but people concerns, and you've been giving me these voice notes, and I read a lot, and I read and I struggle to read the scientific papers that you guys read, so I read different analysis, and then I'll run that past you, and Clive, I've done the same with you, Annemarie, I've done the same with you, and going through this presentation, I'm lost. Like, I get the concept because you've explained it to me, but what I really get is that the science behind it is complicated, and I'll never understand this. So it now comes down to trust. Do I trust you and your peers, which is Clive and Annemarie? Because that even that's it. Like, yeah, I guess that's what I'm saying.
So Steve, what's your understanding of what DNA is or RNA is? What do you understand about it?
This is my point.
Okay. Yeah, well, that's putting me on the point. Right? Okay, so DNA is the information that's held in the cell, and RNA is a protein. Is that right? It's a protein. There's something. Their information. There you go. I've already.
DNA is double-stranded. And because of that, it's highly stable, right? Okay. RNA is single-stranded, and it has a couple of different chemical makeups, but it's basically the same thing as DNA. They're both chemical instructions to our cells to make certain things. DNA is usually long strands of information. RNA is also usually quite long, but not as long. And then you get mRNA, and mRNA is messenger RNA. And basically, it's short pieces of copied DNA or RNA, short pieces of information to code for just one thing, not everything. And basically,
was I right about proteins? At least there was protein in there somewhere.
Well, they code for proteins.
Okay, right.
The mRNA codes for proteins in the cell, basically.
So basically, if you look at these sequences that I've got on the screen, right? 3 of those letters, so CTG, that will code for an amino acid, right? An amino acid is the basic building block of a protein. A chain of amino acids makes up a protein.
Right.
So it's the instructions for a protein, quite literally. It's basically like the binary code that you use in computers.
Right.
So I feel like a slide just on basic terminology for molecular biology right at the beginning is crucial, I think, just to keep people on the same page. And then, and then you've explained the language and the basic DNA biology of the cell, because I don't think people don't know that. I've been posting on my Instagram like what is an mRNA vaccine, what does that even mean, because people don't know, and people have been asking me questions, is it going to change the DNA in my body, and if you don't know how it works, then you could be forgiven for thinking that it's going to alter your genetic material in some way or whatever,
which is a big concern,
which it doesn't.
People have.
And it does. Well, it does a bit, but not in any way that is, that matters, or is any more than any other virus would do if you became infected with a virus.
The mRNA never enters the nucleus, so it doesn't interact with our DNA at all. Our DNA codes for mRNA as well, and the mRNA goes out of the cell, is used by the ribosome to basically make the proteins, but it never goes back in to change the DNA.
Yeah.
So yeah, I think that's a big misconception that people have.
But it does change our bodies in some way. And
it does. It changes the way that our body's making proteins.
Yeah.
But it's
in order to protect against the virus.
Yeah.
But it's not permanent. We have to remember that the reason we don't have an mRNA vaccine for now is it's because it's a very difficult thing to get to work, because RNA is single-stranded, mRNA is very short, and so it's very unstable. So it breaks up very easily. And so before now, we haven't found a way to get it to actually be in our bodies for long enough to make a protein that we wanted to make antibodies against the virus. So it's not going to be a permanent change. It's not going to hang around for longer than a few days.
Okay.
Next.
So what I've taken from what you said, because I actually didn't know that those letters were code for amino acids and everything, but just that you need very specific material, and that the CDC is transparent about what material is needed. It's just publicly available. Is that why this slide was in here?
Yeah, so basically each of these letters codes for, each of these letters is a base pair, and 3 base pairs code for an amino acid, etc., when you look at it from a cell's perspective. But these are synthetic oligonucleotides. So an oligonucleotide is like a fake piece of RNA or DNA that basically attaches just so that you can have that copying in the PCR reaction.
Yeah.
So that's what a primer is, that's what a probe is. So they've basically taken the same sequence as the virus of letters and said that's what we're looking for.
Yeah.
And so based on this, right, so Clive, can I ask, I can pause this before I ask this question, but I'm not going to. So would you, then do you I'll inject this in my body, I'm going to inject this in my son's body? People have concerns, right, about this. Annemarie, same question, you've got a son, that's what people are worried about. Sarah, how do we trust you? How are you getting paid? And everyone's got these questions. Go ahead. Sarah, yeah.
Just to be very clear about what's being shown on the slide, this is not what they're going to inject you with. This is the PCR test.
Yeah.
All right. No, I get that, but I mean, what I'm, I get that about the PCR test, but the question that people have is about when you do the test and then you're using the same kind of processes to create the vaccine, right?
No, not really. The vaccine's a much more complicated process, to be honest.
A more complicated process, but it's still, you're doing this, you're looking at publicly available information, you're doing this coding, you're doing a bunch of science around that, right? It's this kind of level.
Yeah, so it would be longer than the sequences that are shown here on the screen, but it would basically be looking for the same target region. Just the region that, for example, codes for the spike protein.
I wish I had pressed pause now. See, Clive's just rolling his eyes laughing at me.
Yeah,
No,
and
I'm
you
I'm
can
not
kind
at
of
all.
package that in a form that can be actually delivered within the human body, and that's where the difficult part comes in with the vaccines. It's not as simple as just making one of these oligonucleotides and just looking for a sequence.
Right.
But your question is a massive one, and is about safety. It's a huge question, and I think it's probably, for me, probably one of the most important questions about the vaccine. And I mean, we could get to that. I think probably best to tackle that another time.
Another time, yeah.
I think it would be great to tackle, especially since Clive has the expertise in trials, about the process of actually looking at the safety. Yeah. Because essentially, at the end of the day, it's not easy to trust a pharmaceutical company, but it's easier to trust the process.
Right, when you understand the process, yeah. I think, and that's it. I think it's helping people to understand. There's a process with people behind the process, people like you and Clive and Annemarie, honest people with kids. Like two of you have kids. I know, Sarah, you don't, but you've got family. We're just regular people that are trying to find what the right thing to do is, and balance. I always say I'm so glad that I'm not the one in charge of making all of these decisions. It's such a tough set of calls to make around what's going on. So,
sorry.
Oh, sorry. Yeah. Like when I started my study, I kind of went in a little bit with that idea, like, oh, now I'm going to be among scientists and my teachers are going to be, like these savvy people that want me to believe certain things, and they might be paid by big pharma or something. So I have to be, critical thinking. And I went in there just like feeling like a bit of a like undercover agent, like I'm different from you, but you don't know. And it was like super funny to see that these were just normal people, like trying to do their job and like wanting us to really understand what good science is and why. And that was like I really came from that bubble in a way, and I think many people do in my circles, in a way.
Yeah.
That's kind of
it's
how
so
it's
funny,
it's movies. If you think about it, there's always, in a medical drama, there's always someone who's a hero fighting against this big medical company or something like that. And
yeah.
I guess everyone feels like that. They feel like they want to be the person to make a difference and make sure that everything's done right.
Yeah. And also I think science in my practical profession as a physical therapist, science was partly there to inform our decisions, like what are best interventions and stuff like that, and to inform treatment guidelines, which we only had 2 of when I graduated in 2000. Like now there's like 20 or 25. For me, that meant a burden. Like I had much more administration to do. I had to put my individual patients in some kind of protocol that I thought was kind of not personal and individual enough. And the science was used to make me like accountable for what I'm doing. Like I have to defend how many treatments I'm giving, why I'm giving the treatment, if it's effective enough. So that science and that evidence was actually a pain in the ass for me. And many people who switch to more alternative types of professions have this, and they kind of have an evidence allergy. So they see it as a structure that's being enforced on you that is actually a good thing to kind of free yourself of.
To throw off, right?
Yeah. And
I would really, that really resonates. It's extremely difficult to keep yourself up to date with current practice in a responsible way, to be as responsible and answerable to your patients or clients as possible. as you should be. And you know, and it I was just gonna I was gonna comment that I do Brazilian jiu-jitsu, and it's have some pretty interesting conversations in the changing room when we're getting ready for training, and it like people raise, you know, bombshells in the changing room, like we were talking about vaccine safety yesterday, and you know, people were just going, you know, I wouldn't take that, you know, it's definitely doesn't sound safe to me, and you know, and then I explore a little bit in more detail, you know, what do you know about it, and or what do you know about the way it was, how the vaccine's been developed, and usually the answer is not very much. Like it's kind of a gut reaction to it, and then there'll be a big discussion about you know, pharmaceutical companies and again about trust and people's lack of trust in you know, centralized government and also, you know, large corporations. So yeah. It's but as soon as I started speaking, and I was telling them some numbers in studies and trying to give some impression as to the rigorousness of the processes involved to take these molecules through development, through drug development, you know, I think that's the piece that people would benefit from knowing, like exactly how the process is carried out, how robust and rigorous it is, how many people are involved in the process, how much money is involved in the process. I mean, it's billions and billions of pounds pumped into it to make sure that as little harm as possible is done to people. And I think that's what people don't appreciate. Like that's the objective is to do as little harm as possible with these things.
And what's fascinating about that is so many of the people that I speak to would put any like number of other chemical substances into their body where they don't really know where it came from, like mind-altering substances, and they're quite happy to do that. And who knows, they
like
bought it from Joe on the,
that are not quality tested or anything.
Yeah, you know, they're going to go to the psychedelic society, or they're going to get it from Joe on the corner and smoke it, you know, or take it at a club. And like, it's nowhere near as like rigorous, but you know, because I trust Joe, like he got it from his, you know, wherever, you know, yeah, exactly. Even a better example of the kinds of foods that are on the supermarket shelves of unscrupulous companies, you know, that are trying to just sell us, you know, things. So,
can you see that? Can you see that? Jess has just handed. Is this backwards? Do I need
no,
to?
it's correct.
Oh, yeah, I see it. Yeah,
this is in the Guardian. This is in today's Guardian.
Yeah, good pharma. COVID-19 vaccines provide chance for industry to improve its image, right? Yeah. Because you've said it's the Guardian, a bunch of people aren't going to. They're going to be like, he reads the Guardian. We're not going to listen. Tell us something else.
My wife gets the Guardian today, and what do you get tomorrow? The Times, Sunday Times. So
who knows?
Yeah.
Okay. Anyway, so Sarah, should we hand it back?
Steve, just going back to your pharmaceutical. Just going back to your
South African internet has gone down.
Like a techno, it's like a techno tune.
Pharmaceutical industry, chemicals.
Okay.
So ascorbic acid, do you know what that is?
Okay, wait. Going back to the pharmaceutical thing. It broke up over there.
We lost you. We lost all of that.
Oh, so I was saying.
Oh, what a shame.
That is a shame.
Internet connection unstable. You guys hear me now?
Can hear you now, yeah.
Maybe turn your video off, and sometimes the audio works better.
Yeah, I think you're back, yeah. If you turn it off and then turn it back on again.
It's always the answer.
That's
The
it.
devices.
25 years in tech, that's all I learned. Okay,
off
you're
and
back.
Yeah, so just when you're talking about supplements, ascorbic acid is very scary sounding, but it's just vitamin C.
Right.
It's the point I was going to make.
Yeah, it's true, right? Or H2O, like when people when you give them the scientific.
It's the dihydrogen oxide thing.
Yeah.
The dangers of dihydrogen oxide. That's
it'll definitely kill you in the wrong
You
quantities.
can come in. Yeah, it's fine. You can come in. You can come in. What's that? Am I trying to be professional? I'm never trying to be professional. Do you want? Should we have Oscar in the picture?
Yeah, absolutely.
Oh, you changing his nappy?
Yeah.
All right. We can have him on that.
Real life.
Hey.
Hello. Nice to see you.
My family.
There you go. The reason why we do all the things we do. Makes
I have
it
to
real.
go soon, Steve, because I said to Jess half an hour. No, I said an hour, and we've
blown through that.
we've blown it. She's never going to trust me again.
Yeah, trust is a big thing. Sorry, Jess.
Sorry, Jess, says Steve. She's bored and wants to decorate the tree. So
Yeah, I don't blame her. Okay,
So
well,
we came to this point because we were talking about scientists just being normal people. And when you hear Sarah explaining all this, it's just like, wow, there's so much knowledge and work and training and science going into just a PCR test.
yeah.
You know?
Yeah, and you're on slide 13.
Yeah. And then there's like some guy sharing a meme on Facebook that the PCR test is not meant for diagnosis because the inventor said so. You know, and it sounds like that kind of invalidates the whole thing. And then this just gives a different perspective, really. Also, like there's so much work in this. There's so much we know about how to measure test performance. Yeah.
And let's just be clear about something. When he first designed PCR or came up with it, there was none of this fluorescence. There was just looking for the size of the fragment. So it was a lot less specific.
Yeah, it's like a health technology that has evolved. I think it's like 30 years old or something. Like, of course, it's not the same technology than it was back then. And, you know, even if the inventors said at some point that you can't use it for diagnosis, we can test how well it works for diagnosis. We know how, you know, we know the test performance, we know the sensitivity, we know the specificity. All these kinds of things have changed as well, like doing research into diagnostic test accuracy wasn't what it was 30 years ago. So it's not super relevant, maybe even.
Well, should we?
Exactly.
I'm conscious, like, so we're recording. I've taken what should we do time-wise? You know, we're going to come back to this. You know, we're. Do you want to just do like a wrap on the sides, like your main point, and then, you know, we can decide. We can all watch the recording and decide how we do it better next time. And, you know, could I give it to somebody just to edit some of the key snippets? I can try and do it. You know, they are.
I think, Steve, if you wouldn't mind, the last few slides are actually the more basic part.
Okay. Yeah.
So I'd love to just finish, and then you guys can see the point that I'm taking you on.
Yeah.
And then
okay.
maybe what I can do is next time, if we discuss, maybe what we can do is next time you can maybe I can record it as a pre-recorded presentation, and then we can all just pause it and discuss it along the way.
Yeah, that might be
that's
good. That's a pretty good idea, actually.
Yeah,
and simplify it. Like maybe we can just really have a think about simplifying, you know, the message, or even, yeah, getting somebody to sketch it out or something. Yeah. Okay. So here we are at the next few.
Yeah.
And Clive,
So the
if you need to drop
previous
off, then obviously you can, right?
Okay.
Yeah, so the previous slides were basically just showing you how PCR works, how specific it has to be, and then the sequences that are publicly available on the CDC's website. So basically you would take those sequences and go to this website called NCBI GenBank, okay, and you perform a BLAST, a basic local alignment search tool. So what that does is it takes each of those letters that are published on the CDC website that the assay is looking for, the PCR test, and it searches them against a database of basically all the known sequences. Now, if you publish a sequence, this is where it's going to be on this site. So next slide. So you basically, I've taken that probe sequence, I've taken out all the quenchers and the dyes and all of that kind of stuff and just putting in the pure sequence. You plug it into that enter accession number. That's where you enter it and you kind of just decide what you want to search. So I've chosen standard databases and highly similar sequences. Okay, next slide. So now it's conducting the search and it'll usually take a couple of seconds to a minute, depending on how long the sequence is that you put in for it to search. And so it'll kind of go through this and then it'll come to the results. So if you go to the next slide, these are basically what the results would come out as, okay? So you can then filter this to more or less results using the filter tool. Next slide. So these are basically the top 100 hits. Okay, there's not 100 there, but obviously it's a screen capture. And as you scroll down, you would find that all of them say SARS-CoV-2. Okay, so the top 100 hits, it only shows you, and those have all been SARS-CoV-2, right? The COVID virus. So then you can see there's a bunch of different metrics there. So it shows you the percent identity, which is 100%, shows you the query cover, which is 100%, which means that it's basically covered that whole region. And then you're going to go into, because if you haven't understood the previous stuff, then that's very complicated. So if you go to the next slide, this is the graphic summary that it shows you. So showing how it's aligned. And you can see it's all aligned across all 30 base pairs. So each of those letters. So you can look into this in the BLAST search. And then if you go to the next slide, for every single one of those 100 hits, you can actually go into it to look at the alignment to see how they matched. And as you can see, the query is what we were searching for. And this is the whole description of exactly what we were searching for. And then the subject is basically what it found. And as you can see, each one matches with each one. So it's an exact match to the SARS-CoV-2 virus. Okay. And you can also look here at the probability of it binding or not binding based on where the mismatches are. And that also is very complicated. But here you can see 100% match. So if you go to the next slide. Now, this goes back to where you can actually filter the results, okay? And here it shows you the lineage of everything it's found. Everything is SARS-CoV-2. So if we now go to the next slide, we now try to exclude COVID-19. So we put in there severe acute respiratory syndrome coronavirus 2. I've basically just copied that from the taxonomic thing. And then you click exclude. So it's going to exclude all COVID results. And then you would then click filter. So if you go to the next slide, let's see what it comes up with. It's come up with nothing. So it doesn't find anything other than the coronavirus that's specific to COVID-19. No significant similarity found. So that's how we know that the false positives is not picking up other viruses. It just doesn't find them. So does that make sense?
Yep. Even for me. Okay.
Thanks, Sarah. Wow, you had a lot of work, I think, putting this all together. Like,
so this is what us as scientists, before we would even put together the test, this is what we have to do before we even publish it on the CDC website or before we even start testing it ourselves. We have to do this in silico search. And to do this in silico search, you actually have to know a little bit about it, unfortunately. But I mean, anyone can go and do the search.
Yeah, publicly available search, right? So it's transparency. I think that's one of the key things, is this stuff is transparent. Yeah. Okay. Clive, Annemarie, anything to
Can I
say?
read you something? I'm just looking at this article. Can I read you? Because it's like it's bang on topic. I'm gonna read you this. It's very quick. It's quick with my wife singing in the background. Those working in the pharma industry have long been frustrated by its public image. Many are former doctors or scientists motivated by a genuine desire to improve people's lives through medical innovations. This is this bit. Traditionally and historically, public trust in pharma has been comparable to the trust they put in their broadband provider. There are signs of a shift in public attitudes. A recent survey revealed that globally, the industry's trust and like score is at 68.2%, up from 64.9% in 2018. So it's just quite interesting to hear that, and and that trust is that they measure trust in that way too.
Yeah, it's very interesting. Yes, I think I'm going to stop recording, but thanks everyone for listening. Whatever part of the snippet we had you in, so thank you, and thank you, Clive and Annemarie and Sarah, for participating in a very interesting discussion, which we're all learning something. I think we all learned quite a lot. I learned that Definitely. I don't know very much. Even though I read a lot about it, I don't know very much at all.
I learned that I can't convey what I know.
Well, actually on that, because I think you did a really good job, but what Clive said, like, there's going to be people who don't have the basic knowledge to synthesize this. But I can see when you go through the presentation that you've thought out like the individual bits and pieces that someone needs to understand your main point in the end. I would just make that clear with every slide. Like, I've put this in because
of that,
because you need to understand that this and that to be able to get my next point. And I would maybe even turn it around, like start with the main conclusion, like it doesn't pick up other viruses, and a false positive doesn't mean it picks up SARS-CoV while it wasn't there. It just means that it picked it up, but you might not be infectious, which is different. And then kind of weave back all these things that just show how unique and specific these tests are. And then I think people will be following it easier in a way.
No,
but I think you did really
the blast search and then say, what are we searching?
Yeah, I would agree with that. Yeah.
And Clive and Annemarie, did you guys learn anything through this little?
Yeah, definitely. I'm not a molecular biologist.
You know that you're recording, right?
Yeah, I am. I'm going to stop it. I just thought we'd all say that.
Yeah, definitely. Yeah. Also, I thought actually the false positives were, positives that weren't there. I didn't know that it actually wasn't that in this case. And I've heard about lateral flow testing, but I didn't actually understand what it does and why some people say that it is better. But seeing that graph kind of definitely made it clear. So I got a lot out of this. And it's fun, I think, to think about this with other people and like have different views of what's happening and what might be important to share, because I find that just really interesting. Like, yeah.
Clive?
No, I was just gonna say, you know, responding to, thank you, Oscar. Responding to people who say there's a false positive epidemic, like we were referring to earlier. I think you know, this goes part of the way to addressing that. But what do you say to people, Sarah, when they address that particular question to you? You know, is there a false positive? Is there a false positive epidemic?
I don't want to pause it just yet because it's good.
Oh yeah, it's recording. Oh yeah, okay, good.
So yeah, I think my main message there is that it's not detecting the virus if it was never there. It just might detect the virus if it's not there right now, but it was there. And so people might not be infectious right now, but the case numbers are still reflecting the true number of people that have been in some way infected by the virus, whether it's a subclinical infection or a full-blown hospitalized severe case.
I think the media's got a lot of responsibility in all of this as well, you know. So, you know, I wonder if there's some, you know, another person to get on. So we should do a few of these and maybe structure them a little bit more different. I'm going to press pause now, but okay, let me do that.
So, you know, the Mike Yeadon article, for example, like he talks about this a lot. He actually refers to you know, a false positive epidemic, and you know, and then, you know, in the same sentence where he lists all his credentials as a scientist, and so it's potentially quite misleading, I think.
What are his credentials? Just remind me.
Well, I can only tell you what I've read. I'll read it again when you search for him online. So he's a pharma person as well, but he's founded his own company. He has worked. He used to be VP of research for Pfizer, for global R&D.
But is that true?
For a long time, 20 years. Okay, well, is it true?
Because that's
it's in. So you've got the BMJ, the British Medical Journal, have him as he's written he's a former chief scientific officer at Pfizer.
Okay,
but it doesn't say his specific role.
He's worked within allergy and respiratory disease.
Okay, but like what type of, because what I'm getting at is, for example, if you were to ask me, for example, what specific antibodies, etc., a certain company is looking at for allergies, you know, I'm not an expert necessarily in that.
Yeah,
so
sure.
Is someone who's been in R&D and experts in molecular biology, do they know PCR testing and how to design a PCR test?
I think he was dealing with respiratory viruses and inflammation in the body associated with it, and especially cardiological problems.
Yeah, so that's very broad and still not molecular.
No, it's not.
The thing is, I found that an interesting video in a way, because a lot of the critique on everything is so like information that's so easily debunked, and because he does have like a lot of credentials, I was like, oh goody, you know, I can learn something from this video actually. But some of the red flags that I had was like, it's again someone who says I'm an expert, and I have a completely different view of basically what's happening than the whole world does. And I've come to kind of question people that say the whole world has got it wrong, and all these other immunological experts in the whole world, like not just in the UK, because you could say like, you know, I'm an expert on this and I have a different view and that's why I'm not heard. But then basically this would be happening in the whole world. The likelihood of that, the probability is so small that that's actually the case. It's much more likely that he has a point, that there are just better arguments on the other side, or other reasons why we would do this in this way, that he, from his expert experience, hasn't really embraced or grasped or gotten to know. And I think that's just really interesting, because he's saying he has all these credentials. If you miss all that other information, what are other experts in his field saying about this? I would be really curious, what does Sarah have to say about T-cell immunity? Because his whole story leans on there already is immunity, and it doesn't show up on a PCR test because it's from T cells in a way. And then there's also a thing why, if he has all these credentials and he is obviously a scientist, so he would have, connections in science, why go on YouTube? I don't understand that, really.
Or was it definitely him? Was it actually him? I don't know. I mean, I don't people send me all this stuff. I just don't have enough time.
Yeah.
And then I send it to Sarah. Sarah doesn't have enough time, or other people. I send it, Clive. But I saw one and I thought it was the one by Dr. Roger Hodgkins, whatever. That was the one, and I sent it to Sarah. And I watched the original one, and they took it down, and then they changed. They changed the script, and put a picture of a different man up. And that's the one that's now viral.
Oh, that's strange.
Wow, that's really weird. I remember watching the original one and sending it to you, Sarah. And then by the time we got to do it, it had been taken down. And I don't know, there's just so much weirdness out there. But I'm gonna press stop on the recording.
Right.
Yeah.
I mean, just to that, I guess the last thing I say is that the article he wrote about Sage, I think, did you all read that one? I think that's
I read
what
part of it, yeah.
Shall I press stop, Clive?
Yeah, that's fine. We can just have a discussion now.